on.
Great stuff. Thank you very much. I'd like to begin by thanking the session organisers and indeed the whole conference for putting this together. It's been really fascinating so far, and I'm sure it will continue to be for the rest of today and tomorrow. Also, just in case I forget, I'm sure I won't, but in case I do, I'd like to thank the people I worked with on this project and the funders for giving me the opportunity to actually do some experimental work. Okay, so I'm going to talk today about multi-spike rhythms in just a single cell to look at sort of what are the capabilities of encoding memories in a very simple system. So I'm going to present our modeling results and then go on to talk in the second half of my talk about experimental results. But I just want to say that these are quite preliminary because they happened about 4 days before the lockdown was announced in the UK. The labs are starting to open up again, so I'm hoping to collect some more results. But I really just wanted to give everyone a flavour of what we're doing and to seek any feedback that anybody might have. So I'm going to start by talking about a laser system, which may seem like a slightly strange way to begin to talk about neuroscience. But the reason I'm doing this is because it echoes a lot of the ideas that I'm going to talk about. And these kinds of things have been studied in a lot more detail in a laser system. Okay, so it doesn't really quite require an in-depth knowledge of lasers. I'll talk you very briefly through what's going on here. So this is a driven laser system. So the master laser here is driving a slave laser here, such that the slave is phase locked to the master laser. And there's a pulse modulator sitting in between here, so that this can provide perturbations to this phase locking. Okay, so downstream, we measure the activity of the slave laser. So that's what's shown on this screen here. And importantly, the output from this slave laser is split by a beam splitter here, goes down here and is reflected back to that same laser. So this actually is now a recurrent feedback loop, and it has a delay that basically is the time it takes for the light to go from here to the mirror and back again. So what happens, so here's a slightly simplified schematic of just the bit that's important. So this is really just isolating the slave laser system. So you can imagine that there's some external perturbation that I can put in from here, but this is really the feedback loop that's important. Okay, so this represents here the delay, and in this system, the delay here can be tuned because the light actually passes through a fiber optic cable. So if you make the fiber optic cable longer, you make the delay longer. And so what happens then if you give it some external perturbations is you can elicit pulses from the slave laser. And so here the delay, so I'm just going to minimize everyone on my screen. So the delay here is tau. So that's the time it takes for the light to go from the mirror, get reflected back to the laser. And if you elicit pulses in this slave laser, then you can essentially, these can be repeated over time because the light that's coming back to the laser at some delay tau then exhibits this recurrent excitation. So here we can see on the y-axis this round trip number. So essentially you just take a long time series and chunk it up into epochs of length tau, which is the delay, and just essentially stack them on top of each other. So this is in the laser field, they think of this as a pseudo-spatial variable. And so what you can see here is that after sort of maybe 105 maybe round trips, we see a stable pattern of 6 pulses per delay period that basically just goes on forever. So this is a stable, robust pattern. And the idea is that in the laser community, people think of these as memories, so memories that can be stored by the laser system. And in actual fact, that as the pulses are triggered, we can store a variety of patterns and they coexist, and these are all stable. So we can have patterns with 1, 2, 3, 4, 5, or even 6 pulses per delay period. And the only real thing that we need to have here is that there's some excitation and there's some delay, and the delay needs to be sufficiently long so that we kind of escape this refractory time. So really the question here is, what are the patterns that coexist? Where are they in parameter space? And really we're asking about the capability of the system to store memories. Okay, so that's in a laser system. Of course, we're more interested in neurons. So let's think about neurons. And of course, this is a slightly more traditional thing to think about memories in. So really, what is the capacity of a neuron, a single cell, to store memories? So this is the analog of the laser system, but in a cell. So here we can see the cell body. And of course, as we know, neurons are actually spatially distributed. So here is my axon here. So activity at the cell body will be transmitted down the axon, as I've shown here with these arrows. So if I elicit some spikes at the cell body, I will see them a little bit later at the axon. And I'm just assuming here that the shape of the spikes is the same at the axonal bouton. So there's no attenuation of the signal. But of course, the fact that this is spatially distributed induces some delay, which again, I'm going to call tau. So the tau here is now the time it takes for a signal at the soma to reach this axonal bouton. So this is one cell, and of course, in a neural network, these guys here would be coupled to the dendrites of some receiving cell. But what we can do in this example to emulate the laser experiment is to couple the cell to itself. So now I've just basically bent the axon back round, and now I can see that there are these sites where the cell projects to itself. And now if we give a signal at the cell soma, it will travel down the axon, again with this time period tau, and then there'll be some synaptic activity here. And if this is sufficiently strong to induce a postsynaptic response, then we can hope to get this recurrent activity in this same cell. So this is what I'm going to call the ORTAPS model. And here is a really simple example of a memory, or pseudo memory, if you like. So in this experiment, this is just simulated data. So I give some applied current at some time, I elicit one spike, and then I don't give any more current. And now what happens is the postsynaptic activity is here induced in that same cell, and activity builds up, and after maybe 3 spikes, I'm back on the same rhythm that I started with. So here is an example of a successful encoding of a 1-period rhythm. So these are the kinds of things that we want to try and understand. So I'm going to begin now by talking about the modeling framework which we use to do this. So this hopefully will be familiar to people who study computational models of neurons. So this is the classic Morris-the-Car neuron. We chose this model because it exhibits quite a lot of the kinds of behaviors that we want to see, and in particular, it's tunable to different kinds of sort of bifurcation scenarios. And it's really nice and simple because it's 2D, and because it's 2D, we can analyze it in quite some detail. So here's the standard Morris-the-Car model. So we have some voltage, which is the thing that we typically plot when we talk about action potentials, and some recovery variable W. And for those that are interested, here are some steady state functions and time constant that go along with this model. This model, of course, is not coupled to itself. So we introduce this ORTAPS model. So S is some ORTAPS induced current, and I assume it has a very, very similar form to W. So there's some S infinity, there's some asymptotic steady state, there's some time constant S, which in this model, we're going to assume that the synaptic transmission is instantaneous. So there is a delay term, which is the delay between when the signal is felt at the axonal bouton versus when it is elicited in the soma, but the actual transmission of the activity across the synaptic cleft is instantaneous. Okay, so we can replace S with just this asymptotic value here. So here's the model, and what we want to know is about, well, what are the memories that we can store in this model? So the classic thing to do is to do some bifurcation analysis, which we can handily do in DD-BIFTOR. So here is an example of when there is no coupling. So kappa here is my coupling strength, and tau is my delay. So if kappa is 0, there is no coupling. So this is the classic bifurcation diagram for the Morris-the-Car model. And we can see this S shape here. So this is an equilibrium, and we can see a hot point over here on the right. So this is a bifurcation diagram that will be familiar to people that have studied the Morris-the-Car model and the current injection. As I introduce the coupling, the self-feedback, so as I increase kappa, we see hop bifurcations begin to accumulate. Okay, so they start off being quite close to this upper hop branch, and there's one over here. And as I continue to increase it, all of these guys shift over to the left. Okay, so by time kappa equals 30, there's lots of hop bifurcations on this curve. And this can be summarized here. I've just taken all of these 1D slices up to some notional value of kappa and just stacked them. Okay, so you can really see that these hop bifurcations, the rightmost one is shifting to the left, but also they're getting more and more spread out along this axis. All of these hop bifurcations here are of the subcritical type. However, they do elicit periodic orbits that stabilize via Folder limit cycles that I'm going to talk about in just a minute. In a similar way, we can do a continuation for a fixed value of kappa, but changing tau, which emulates what was done in the laser experiment. And again, we see something very, very similar. So remember here, kappa is chosen to be 60. So even at 0 delay, the cell is coupled to itself just with no delay. But we still see a very simplistic bifurcation scenario. However, as we increase delay, then we start seeing this collection of hops. And this sort of mirrors what I said earlier, that the delay needs to be long enough for us to get this memory kind of phenomenon. So we want to be in a situation where the cell is excitable, not oscillatory. The results that I'm going to talk about don't depend heavily on that. We can still compute similar things if the cell is in an oscillatory state. But for reference, we set the cell to be here. So this red plane is where we choose our current density. So the cell is excitable, and that means if we elicit pulses in the undelayed system, we would, the non-coupled system, we would elicit one spike and then come back to rest. So here is an example of what happens then if we fix the value of kappa, and we now, rather than changing the current injection, we change the delay. So each of these curves that we're seeing here, these are branches of solution that are elicited from a hop. This gray point here is the hop, and we can see that we do see lots of stable branches. The green here is, the thick green is a stable branch. We see lots of stable branches that all correspond to different firing patterns elicited by this same cell. Moreover, we can see that actually very little information is conveyed in the amplitude of this spiking behavior, which is precisely what we expect in neurons. Typically, the firing of a cell is an all or nothing kind of event, but a lot of information is encoded in the period. So we can see here that each of these different patterns has periods that are very well separated from other patterns. Moreover, if we then look at what happens in the two-parameter bifurcation diagram, so now we're changing coupling and the delay, we can trace out folds of limit cycle bifurcations that basically take us to regions where we can express different numbers of stable patterns. So the number of patterns that we can store is indicated here in these little patches. So here we have 1, then 2, then 3, then 4. And here, in these panels here, we're just taking a slice at kappa equals 60. If we actually look at these solutions, so if we look now, we fix the delay to be 400, and we just look at solutions D and F here, we can indeed see this. So here are two solutions, D and F. We're plotted here. This is twice the delay period, just because it looks a bit nicer. So here we can see an example with 2 spikes per 2 delay periods, so 1 spike per delay period. And here we can see 2 spikes per delay period. And the point is that these solutions exist, these coexist, and along with a whole bunch of other solutions that you can see on these other branches here. The other thing that I want to say is that if you look at this solution here, so these are the Floquet multipliers of this solution. And you can see here there's one that's really, really close to the unit circle. And in fact, we see this for a lot of different unstable branches down here as well. And what that means is that even though these are the asymptotically stable solutions, there are lots of other solutions that are asymptotically unstable, but that we can stay close to for a really, really long time. So if we do an in silico simulation where we now give 2 pulses rather than just 1 pulse, in some scenarios, so the 2 spikes elicited by these 2 initial pulses, but then what happens is that the second pulse then falls away. So actually the solution that we get in the simulation is just 1 spike per delay period. Conversely, if we slightly increase the distance between these 2 pulses, we can the second spike can persist, and we can get solutions that look like this. And these will persist for a really, really long time. You can see here that this is number of delay periods, and you can see this is what, 10 to the 6 almost. Ultimately, of course, it will give way to the stable solution, but it takes a really, really long time. So these have all of the hallmarks that we might think of as memory. So the ability to store different kinds of patterns, but that decay over some quite long time scale. So this is basically a blueprint for understanding when this ORTAPS model can store memories. So a sort of sensible question to ask, well, does any of this actually happen in a real cell? So now let's go to some experimental results. And I guess some of you may well be familiar with the experimental technique, but as a refresher to those people and as a very, very brief primer to other people, I just thought I'd spend a few minutes just talking about how the experiments actually work. So this is due to an experimental technique called patch clamp. And the idea is that what you do is you take an electrode and you put it in a little pipette like this. So you have one electrode that sits outside of the cell, and this is sitting in some saline solution that tries as best as possible to emulate what the ionic composition in your brain is, basically. So we set up some solution that matches cerebrospinal fluid. The other electrode sits in this solution here, which is now, this composition is matched to be what is inside the cell. And so you land it on the cell, a bit like landing on the moon, and then you sort of suck quite hard on a little tube and you rupture the cell membrane. So now you have sort of a contiguous solution here between the stuff that's inside the pipette and the stuff that's inside the cell. And more importantly, the electrode is now inside the cell. So we've got one electrode inside the cell, one electrode outside the cell. We can measure the potential difference across the cell membrane. And then we can track these voltage fluctuations that we know as action potentials. This is not what I actually did in these experiments. because that's actually a bit rough on the cells. They don't really like it if you blow big holes in them. So instead, I used a method called perforated patch clamp, where we actually put an antibiotic in the cell. And what this does is it just lyses really small holes. So we get a much stabler long-term recording, which is useful if you're trying to track things like memory. Here's an example. So here's my pipette with my electrode sitting inside it. So I landed it on this cell here, and you can record activity that looks like this. So these are, spikes that we know and love. So there's the voltage of this cell over a 5-second window. Okay, so that's half the job done. But of course, these cells are sort of isolated, okay? And actually the probability of finding a cell that is coupled to itself is pretty small. So actually we rely on a technique called dynamic clamp to get over this. So what dynamic clamp does is it takes that signal that I'm recording from the cell, it amplifies it, and then it runs that output from the cell through a mathematical model. So this is just done on a computer. And the computer here essentially allows the direct interfacing of any mathematical model you want to give and a cell. So the model on the computer essentially computes what current you should inject to the cell to encapsulate whatever description you put in here. And then it basically takes that current and injects it back to the amplifier, and then the amplifier sends it to the cell. So this has been around for a few years now, and it's typically used to either virtually insert or remove specific ion channels. If you get your model bang on, you can also use it to modify specific ion channel properties, such as timescales. So it's really, really nice because it's quite a clean experiment if you really believe in your model. So sometimes the pharmacological experiments or the gene editing experiments can give you effects that you weren't really expecting, but this really will just do whatever the model sets. So in our case, we're going to use it to couple the cell back to itself. So we're going to use it to essentially make this green loop here. So let's go back to the model and think about what it looks like. So here is the Morris-Le car with the AuTaps. So now if I color in the different parts of the model, so the blue bits is basically the model of the cell. So this is what the Morris-Le car model did. And of course, any kind of Hodgkin-Huxley model will look like this. So I'm just going to leave this to the cell. I'm not going to impose any other dynamics other than that with regards to the intrinsic properties. And this red term here is the AuTaps model. And this is what's going to be handled by the dynamic clamp. And just to show you how sort of the workflow works, just to repeat what I said earlier. So we measure some activity from the cell. Because now we're doing some delayed system, we have to store the history in a buffer. It's actually done on a little chip here, which I'll show you in a little bit more detail on the next slide. There's not that much memory on this chip, so I actually use a little circular buffer here to store the values of the voltage in the past. Then this guy here computes this autoptic current, computes the current, and then rescales it, and then puts it back into the cell. And this loop just essentially repeats. It just goes on and on and on and on. And that essentially will do the integration of this term in this cell for me. So here is what it kind of looks like. I guess one of the things that I find really interesting about this is, this is just built on a breadboard. I haven't even soldered anything in. So there's a paper that came out in 2017 that really shows how you could make dynamic clamps really, really cheaply. Before then, they were, you needed a whole different computer, a whole different setup for recording. But this is a really cheap, really efficient way to achieve the same kind of result. It's really fast. So this little chip here is it's called a Teensy 3. Well, this is a Teensy chip. This is a number variant 3.6. But it's really, really fast. It samples pretty robustly at 100 kilohertz. It doesn't have that much memory on it, but it has enough to do what I want to do. And importantly, it's got a lot of analog inputs and outputs. So you can use this to control quite a lot of properties in the cell, or even couple other cells, which I didn't do, but would like to do in the future. So this is really nice. It runs in Arduino, so it's basically a little bit like programming in C or C++. I wanted to exhibit a bit tighter control over the cell when the experiments were actually going. So I wrote a little Matlab controller for it, which looks like this. So if anyone's interested in using that little Teensy thing, you can download this controller from my GitHub here. And I really like it because I use Matlab to also do my electrophysiology recordings. So in support of open source software, you guys can check out the Symphony here, which is a great Matlab package for electrophysiology recordings if you see fit. Okay, so that's all I wanted to say about the setup. So let's go to the results. And I stress again, these are reasonably preliminary, but I think they illustrate the point quite well. So here is an example of what happens if I just patch the cell. I'm injecting no current now, so this is just spontaneous activity. And we can see pretty nice, robust tonic spiking activity. This is not quite what we want, because if you remember in the model, we set ourselves to be on the other side of the bifurcation. So our cells are excitable, so we can just inject a little bit of hyperpolarizing current to essentially put us on the other side of the bifurcation. So this looks like this. So now everything is set up for us to actually run this experiment. So here is the actual experiment. So now you can see the red here is the applied current that is sort of standard electrophysiology experiments. So you just give some square wave pulse or something else. But then I switch it off. So I give it a little pulse to begin with, and then I switch it off for the rest of the time. And in gray here, we can see that the current that is being injected by this little chip here. So this is the dynamic clamp. This is the autoptic model. And what we can see is that even though I don't apply any more current, that the spike shape builds up, and it's actually pretty robust. So this actually happens after just one spike, and then this persists essentially indefinitely. So this is the example of this simple encoding, this simple one spike to one delay period that I talked about right at the beginning. So that's the one spike experiment. What about the two spike experiment? Okay, so this was the next thing that we tried. So this is kind of the same kind of picture we saw before. So round trip number on the y-axis, and then I just renormalized everything so that my delay period is 1 here. So if we try and give two spikes that I'm going to color code with a little orange arrow and a little green arrow here, if we get the delay period wrong, or the coupling is not big enough, then we cannot elicit recurrent spiking activity. So here we can see the two spikes that they're elicited by these two initial pulses, but we don't see any activity after that. Okay, so if I retune it a little bit, so now my coupling strength is high enough, but my interpulse interval is a bit misspecified. So you can see here that the spike that's elicited by the orange arrow initially starts high, then it decays, and then it disappears. So this does not repeat indefinitely, but the spiking activity generated by the green pulse does. It builds up precisely in the way that we saw in the laser and in the model. So after 3 or 4 spikes, it's reached its maximum amplitude, and this will just stay now for a very long time. And as you can see here, that this is actually even more stable than the laser. So I elicit some spike down here, and then it locks on pretty tightly. So this is the one spike to one delay period solution. If I change my interpulse interval, the interval between my two pulses that I apply, I can generate this 2 spike per delay period solution. So now both the spike elicited by the orange and the green arrow build up over time, and we can see this 2 spike per delay period solution here again, which looks pretty robust. I would imagine that the model predicts that over time this should give way to a solution where these spikes are separated by essentially a phase pi. This experiment didn't go on long enough to check that, but that is something to check once I get back into the lab. But essentially all of this is evidence that these solutions do exist, they coexist and are both stable. So these are both solutions for the same parameter values, the same value of kappa, the same value of tau. And I actually even tried doing a 3 spike solution, which is not shown here, but that also gave me something very, very similar. Okay, so that's essentially all I really wanted to say. So just to summarize that, so memories, and by memories I really mean persistent spiking patterns over some delay, can be encoded in these just a single cell with a delayed autotaps. The system is multistable, so we can support different kinds of rhythms under different variation of coupling parameters, and in particular the delay period. So really, to me, this is I think of these as little memories, little packets of memories that can be stored by just a single cell. And as well as the stable branches that we see, there are also loads and loads of unstable branches that are only weakly unstable. So we can stay close to them for very long periods of time. And actually, I think there is evidence to suggest that there are other stable branches of solutions that don't have equispaced pulses. So there are probably even more stable solutions than we've looked at here. And in the future, we'll be trying to unravel a little bit more of that, exploring what happens with different underlying bifurcation structures. And more importantly, I think to the neuroscientific community, what happens when you couple multiple cells? How does the sort of scaling of this multi-stability, how does that change when I increase the number of cells? So with that, I just want to thank the people I worked with on this. So Krasi and Bernd, who provided a lot of the some interesting discussions and input about how the system works, and Piotr and James, who helped out a lot with the DD-BIF tool, and Andy, who helped me build my rig in the first place so that I could do the experiments. Thanks very much.
Thank you very much, Kyle. Perfect timing. Excellent talk. Yeah, we see some comments in the chat.
I think Jonathan has a question for Kyle. I'll unmute him so he can ask him directly.
Yes.
Thanks, Daniele. Thanks, Kyle.
Hi, John.
Nice talk. Hi. So thinking in terms of memory, how do you interpret this sort of multi-stability? Do you interpret these spikes when you have more than one spike sort of per period? Do you interpret them as part of the same memory or a sort of multiplexing? Can you comment on that?
That's a great question. I guess I haven't really thought too much about multiplexing. I guess it could be. I mean, in the laser community, certainly they do play these kind of games where they start on a pattern, a rhythm, if you like, so multiple pulses per delay period, and they show that you can switch on and switch off different spikes within that. So in that regard, I guess you would think of them as multiplexing, because you can think of these extraneous perturbations you get as coming from different sort of different afferent inputs. In terms of the single cell model, I tend to think of it more as the memory is the whole spike packet itself. So you know the memory is essentially encoded in the inter-spike interval that we see in this delay period. But I haven't really explored that very much. So that's sort of my hypothesis at the minute.
Okay, thanks.
Daniele?
Yes, Kyle, very briefly. So it seems to me that the computational part used Maurice Lecar, but I wonder, you know, and of course this does a good job in reproducing what the cell does in this context. How far can you push that? Is there an even simpler model, or what it makes it work of the Maurice Lecar?
That is another great question. I think the key thing is, I guess, if we think about it from a mathematical context, probably what's more important is understanding what nearby bifurcations are in the non-coupled, non-delayed system. And with that in mind, a lot of the dynamics that we're seeing, well, if I can show you, actually I can go back to the actual bifurcation diagram, which is somewhere. So I think in this particular scenario here, which I think is the closest to the experimental data that we saw, a lot of this is being organized by behavior near homoclinic bifurcations over here. So I think any kind of model that supported a sort of homoclinic bifurcation or a SNCC bifurcation, we would probably be able to see similar kind of results. I will say that we also looked at the scenario of dynamics near a hop bifurcation, and we saw similar but different results. So we could still see multi-stability. Sorry, I'm just going to a slide here, which is right at the end. We did see multi-stability. So again, here, the number here is reporting the number of stable patterns that we see in each of these regions. But now the bifurcations that switch us between different solutions are different. So there's a fold here, as we saw before, but there's some hop bifurcations, there's some torus bifurcations. So the scenario is a little bit more complicated. But I would say that any system that supports a homoclinic, for sure. Systems that with hops, I guess so. I mean, I guess probably you'd want to know about whether things like integrate and fire models can do it. I'd say probably, but I'm not completely 100% sure.
So can we move to another question, which is actually, I think, very important. On your last slide for the experimental data, can you comment on whether this is average over cells, or it's the same cell, or yeah?
So these three experiments on this slide are all in the same cell. The one on the previous slide was a different cell. So I think in total, I tried maybe 4 or 5 different cells and saw quite similar things. I mean, I think that is one thing that I think I would like to stress is that, you know, these data are not highly tuned and sort of this was the cell that I got it to work for. It was reasonably easy amongst the sort of 4 or 5 cells that I tried. Of course, the cells have a different intrinsic properties. So the results in terms of exact timings and exact parameters are slightly different. But no, this is all in one cell.
And one more related question, final one, is what would be the, from Victor Matveyev, what would be the metabolic limit on spike per delay per unit time?
That is another great question. The short answer is I haven't really thought about it. Obviously, there will be one. I think a great thing, I think, for us to extend the modeling, and we could extend it to include some metabolic costs of actually eliciting all of these spikes. And we can try and find a limit there. I would hope that we see the limit in kind of a smooth way and we don't just fry the cell. But it's a great question. I haven't really thought about it too much, but I think it would be great to incorporate it into the modeling into the technical cell.
With that, I would thank again Kyle. Great talk, Victor says. And we'll move to the next speaker.
Thanks very much for all the questions.
Thank you.